Well, welcome to the October 3rd meeting of the IMAG-MSM Working Group on Multiscale Modeling of Viral Pandemics and the GLIMPRINT mini-seminar series. As always, I need to remind people that these meetings are live streamed on YouTube and available for subsequent viewing, so please keep that in mind as we go forward. Tom Helikar and I are co-leads of the group, along with Reinhardt and Jim Sluka and Lorenzo. And we are always available to take your suggestions and comments, anything we can do to be more useful. We have a variety of communication channels, and they work better when people use them. We have the glimprint.org website, which could use some love. So if you'd like to help with that, we would appreciate it. We have a Slack channel. We have a Twitter handle, MSM Viral. We have the classic IMAG MSM wiki page, a LinkedIn page, and the YouTube channel archive of talks. If you have a publication to share that you would like to have promoted on the IMAG site and also on the GLIMPRINT site, please send that to Jim Sluka. We'd be delighted to make sure that everything that's being done in this area gets the attention that it deserves. You can post job announcements on the GLIMPRINT site if you would like, workshop announcements, meetings, publications, funding opportunities. All of those can be put up on the GLIMPRINT site or sent to Jim Sluka to post. Reinhardt has been looking for faculty in computational biology. Please contact him if you have any interest. Jason Shoemaker has been looking for students and postdocs on influenza infection. Please contact him if you have interest in those positions. There's a new RFA for the NSF Pathways to Enable Open Source Ecosystems POSE Awards with a deadline of January 14th for next year. That's a very interesting program because it really funds infrastructure and management more than science. Typically, those kind of projects are hard to fund with classical means. There's going to be a workshop in a year in Bordeaux, France on virus dynamics. If people are interested, you have the contact information here. Coming up in a few weeks, there's going to be an NIH Artificial Intelligence workshop on precision medicine of diabetes and chronic diseases. You can go online or in person. I know Grace was talking about that at the IMAG MSM meeting the past few days, and it sounded quite interesting. Please take a look at that if you're interested. There is a NOSI on systems modeling of infection and immunity across biological scales. This first deadline, of course, is coming up this week, but there are going to be standard due dates on submissions for that on the regular NIH due dates. We have a good selection of speakers coming up for the next few weeks. If you would like to suggest a speaker, please do so. As always, suggest yourself, suggest your graduate students or postdocs. If you've spoken before and you have new material, feel free to come and give us an update. If there's material you've got on other topics that you'd like to present, do that. If there are people that you would like to hear, experimentalists, industry people, infrastructure, model sharing, artificial intelligence, anybody you would like to hear as part of this community, please do let us know. There's a Google form to suggest speakers. You can email me, jglazier@gmail.com, or Lorenzo, or Jim Sluka, and we'll do our best to get the lineup that you're interested in on the agenda. Are there any other announcements before we break? We are scheduled to have a steering committee meeting tomorrow morning at 11. If you think you should be on the steering committee and you haven't received an invitation, let me know, and we'll make sure that that gets sent to you. Any other announcements? Well, I don't want to take any more time from our speaker today. We have uh, Yoram Vodovotz from University of Pittsburgh, who's going to be talking to us about critical illness digital twins, insight from the fusion of data-driven and mechanistic modeling. And I'll turn the screen share over to you.
Okay, again, thanks, Jim. Thanks to all the organizers of this very important working group for inviting me. Today, I'm going to talk to you, as the title says, about applications to critical illness of digital twins. And really, it's a broader discussion of the role of modeling inflammation in this context. I would like to start just by acknowledging funding from NIH, DoD, and DARPA. Note potential conflicts of interest as a co-founder and stakeholder in a company called Immunetrics. My role is the scientific advisor for another company called Anuna AI, and then also, although not directly relevant, but my role in as editor-in-chief of Frontiers in Systems Biology. I also want to acknowledge the role that my Department of Surgery as my primary home department, and now the 20th anniversary of the center, a virtual center that I helped direct called the at the McGowan Institute for Regenerative Medicine, the Center for Inflammation and Regeneration Modeling. I want to give a special, although I'll have an acknowledgement, I want to give special acknowledgements to Tim Billier, who's the chair of my department and a long-term supporter. It's my 25th year in that department. Gary Ahn, who most of you know, Gary Ahn, who most of you know, Steve Chang, the CEO of Immunetrics, now part of Simulations Plus, my core team members, Derek Barkley, Jinling Yin, and especially Faith Naldahibi. Ruben Zamora, who's done the yeoman's job of really running my group, and a lot of the some of the interesting computational biology that I'll talk about, as Greg Constantine, a statistician and mathematician, and then some very, very talented trainees, Andrew Aboud, Lucas Shimone, Nabil Ajar, and most recently Ajti Shah. And I'll mention them somewhat more specifically as the slides come up. So my talk is about critical illness, but critical illness is really intimately intertwined with inflammation, which again, if you've heard Gary Ahn speak, I'm sure by now you're sort of sensitized to that. It's important to go over what it is that inflammation is all about, at least from my point of view. Inflammation is communication. It's the way the body tells itself that things are changing, either from the outside in the context of severe injury or infection, or from the inside through the process of degenerative diseases or autoimmune diseases. It's necessary for proper healing and regeneration. It's a key component of the response to stress. It's highly intertwined with the stress response. And in this intertwining, it's intertwined with the neural system, coagulation, essentially every aspect of organ function, largely because every organ, every tissue has within it not only the parenchymal cells that do the structure or function jobs of that particular tissue or that characterize that tissue, but intertwined among them are resident inflammatory cells that essentially set up for the ability to have local inflammatory responses. And then intertwined with all of that is an interaction with the nervous system, which is very important in regulating inflammation. And I'll touch upon that in my talk. So this is a highly complex and redundant and interconnected system. It's almost a prototypical complex system. It has all the characteristics, nonlinear trajectories, a high dependence on initial conditions and feedbacks and thresholds. And I probably don't have to explain any of that to this particular group. And all of this manifests as dynamic patterns, especially for thinking in terms of omics, et cetera. And ultimately, all that stuff I said up until now is really not in any way detrimental. But as we all know, inflammation becomes dysregulated in many diseases. And our team especially has had a focus on perhaps the largest and fastest manifestation of that derangement, which is in the context of critical illness, which is the response to severe infection or injury that manifests as multiple organ dysfunction. And ultimately, this is the point. It's a puzzle. Inflammation can be both good and bad. There's a lot of effort to try to modulate inflammation. A lot of that has not worked very well, largely, I think, because of this dual nature of the inflammatory response. In our particular group, we've focused on inflammation across what I would call the life and health span. So at the young end of the range, we've done a lot of work in pediatric acute liver failure. The bulk of our work has been kind of in the prime of life, if you will, focusing on the response to severe trauma, sepsis, COVID-19, and wounds, and also in other specific instances, some other particular disease states. And then less so, but have done some work in the context of aging, the responses of aged trauma patients, and also healthy versus unhealthy aging. The goal is, and the message is that it's sort of a mindset that's assisted by a computational tool set. And the goal is to unify data across both time, as many people do, I'm sure most of you, using dynamic modeling, but also space, meaning tissues, which is what I'm going to, and organs, which is what I'm going to focus on a lot in my talk this afternoon, and using both data-driven and mechanistic modeling, and then ultimately trying to use that for rational control of inflammation using AI. And so again, don't really have to explain this to this particular group, but again, it's important to get our assumptions straight, thinking about how it is that we might do this type of integration. Data-driven models, we're all familiar with them. They're association-based. They may suggest principal drivers and dynamic networks and could hint at mechanism, but they're not inherently mechanistic. This used to be a pretty simple statement for me to make, that they make limited use of prior knowledge, except maybe in the context of, pathway analysis software or things like that. But the reality of it is now, with the advent of large language models, foundation models that basically are based on the sum total of what can be found online, and much that is in private data repositories, now begin to blur the line between what is a data-driven model versus what is a mechanistic model. But of course, as we all know, these types of models typically require large amounts of data, and frankly, most of the data we obtain in the biological context don't truly fit the definition of truly big data, in the sense that we may get lots and lots of variables, as in omics, but we typically only have tens to hundreds of patients or subjects at the most. We may do a lot of single cell work in what looks like thousands and thousands of cells, but the reality of it is they're coming from 5 or 10 subjects. So it's very important to realize that a lot of the tool set of data-driven modeling may not be fully appropriate for the kinds of biological questions we want to ask. And ultimately, the biggest limitation, to my mind, is that you're really always predicting within the training data. And yet often, as we all know, these kinds of approaches are used incorrectly to predict outside the training data. Of course, on the other side, we have mechanistic models. Again, I think most folks on this call are quite familiar with that, so I don't really have to explain, but it's important to show the contrasts and similarities. So of course, these kind of models are based on principles, in our case, biological principles, or some other mechanistic or quasi-mechanistic interconnection, and can suggest non-intuitive aspects. This, by definition, requires extensive use of prior knowledge. But as I said just now, in the era of LLMs, I don't know that you could say that mechanistic models are truly leveraging more prior knowledge than a data-driven model that's built off of an LLM. What we do know is that this allows for explanation of emergent phenomena, and again, there's a lot of press and hype around LLMs with the appearance of emergent phenomena, but I would argue that what you're seeing there is just the fact that it's been trained. These types of models are trained in such a large amount of data. Of course, this is in its classical form. Mechanistic modeling is typically more difficult. It requires a lot of expert knowledge, usually teams, to put these types of models together. But ultimately, that effort is rewarded by the ability to predict outside of the training data. Again, this is just a 35,000-foot view of the pros and cons. There's many others that, as you get down into the weeds of how it is you actually carry out these types of models. But ultimately, what we want to do, if we're thinking about especially the application of digital twins, is to think about how it is that we could leverage interactions among these types of approaches in order to get the most benefit while minimizing the challenges. Again, I don't have to tell most of the people on this call, but as we're transitioning now into this era of digital twins, which again are very much based on computational models that are personalized in some fashion or another, there's been this report from the National Academies. Some of you participated in that. And this was the definition that was put forth. And I don't need to read that out for everyone, but I'll focus on the aspect of the idea that this type of model is considered to need to be dynamically updated with data from its physical twin. So the question about that is whether, in fact, in the context of biology and medicine, any such application actually exists right now. I think that a lot of what's out there, including work we have done, is about, it concerns personalized models in one way or another, and maybe sets the foundation for updating and essentially recursively coming back to carry out some action or to do some sort of a new prediction of state or whatever it is that the application is. But I don't know that I've seen, and I'd be happy to be corrected, and it may be that it's happening behind corporate firewalls, but I've not seen any application where this definition of a digital twin is actually being carried out. So in that regard, I use, I will use a kind of digital twins in quotation marks to say that we made a patient-specific model, the first one, I think, of inflammation back in 2008, leveraging by what that time had been about 4 years worth of work in modeling inflammation, either in an abstract sense or in the context of in silico clinical trials. It's, for reference, it's the 20th anniversary of our first paper on this in critical care medicine, simulating the clinical trials for anti-TNF antibodies in the context of sepsis. So we published this paper in 2008 with our colleague Kitty Vertolini at the McGowan Institute. And for people that are not aware of this, so on the one hand, this particular application is itself a clinically relevant one, which is acute vocal fold injury is something that happens to anyone who talks loudly or talks a lot. And I'm, of course, one of the people in that camp, but it's more relevant to singers, to other types of people that are making a living off of their voice. But also there are other means by which vocal fold injury occurs. But more importantly, it's also a pretty fantastic application of a system where you can get IRB approval to carry out an injury on a human tissue and then obtain data from those volunteer subjects and carry out interventions, right? So all of this is very difficult to do if you were thinking about true severe trauma or sepsis or some other clinical application. So what we did in this study, we used an agent-based model to simulate phonation injury to the vocal folds in humans. And these simulations were able to reproduce trajectories of inflammatory mediators that were assessed using a laryngoscope in these subjects. So you could look at laryngeal secretions, and we had data up to 4 hours post-injury. And what's important is we use these personalized models to predict the levels of these mediators at 24 hours. Moreover, we could carry out in silico trials of several different interventions. So spontaneous speech, meaning continuing to do what you're doing, which generally makes things worse. Total voice rest, which is the typical thing that was being prescribed at that time. Or this idea of resonant voice exercise, which is a kind of rehabilitation exercise for your vocal folds. And so we could do these trials, and we could, in fact, show that we could predict individual responses. And the question was, could we do the same thing for something much more difficult, much more involving multiple organs, multiple tissues, systemic inflammation, which is critical illness? So that was kind of our next step. So we were thinking about critical illness. That's been a huge focus of our group, and specifically the idea that we can, of course, look at the systemic circulation. You can obtain many different types of data from the systemic circulation, and there are other pros. So critical illness by itself is intrinsically tied to systemic inflammation. We can make the argument that inflammation, when it is localized to individual tissues, is generally within a beneficial regime, and then it hopefully resolves, but that when the insult is severe enough or under a particular set of, say, genetic predeterminants or other types of set point differences, it spills out into the systemic circulation is a key hallmark of critical illness associated with pathology and eventually mortality. So yes, on top of that, it's convenient that the systemic circulation is easily accessible. But less clearly appreciated pro as well is that because we can get cells from the blood on top of, say, inflammatory mediators that we can measure, we could get DNA from those cells, and we could assess single nucleotide polymorphisms, or SNPs, that are associated with a propensity to produce either higher or lower levels of particular inflammatory mediators, and we can connect that to parameter values in these personalized models. And so we did that in the paper that we published in 2015 in Science Translational Medicine that we call Trauma in Silico. But importantly, we also showed that you could do the classical thing that I just said that mechanistic models are terrific for, which is we calibrated the model on 30-odd trauma survivors. And yet, in large-scale simulations of these calibrated models, we predicted essentially the percent mortality that was found not only in our 500-odd patient population, but also in other large-scale studies of trauma. So we could do the thing that data-driven models really can't do, which is we could predict death having calibrated solely on life. Since that time, we've also taken on various data-driven approaches to characterize the response to severe trauma. For example, this paper that I'm showing here, and then collaboratively, especially with Tim Billier, have done multiple omics-related studies in the same context. So all that's well and good, but the main con is that we're not really getting insights into organ-specific impact. And again, as I just said, critical illness is characterized by multiple organ dysfunction or cascading failure of organs. And so this is a major, major barrier that needs to be overcome if we are to truly make an impact on critical illness and via the means of a digital twin. And so that's when we picked up this next phase of the work, which is to look at organ-specific responses over time. In this case, we're mostly focused on protein-level inflammatory mediators assessed using the Luminex platform. And of course, the idea is not just to look at individual organs, but to look at the dynamics and how it is that there's interaction across these tissues. And although I'm showing a cartoon human, the reality is, of course, for practical and ethical considerations, that we did most of this in experimental animals, although not all, as I'll discuss in a second. So a lot of that work has been published. Looking down here, our first study in 2018 looked at a mouse model of sepsis, which is really not sepsis, but acute inflammation using endotoxin in mice, bacterial endotoxin in mice. And this paper was a companion paper that came out a couple of years later that expanded upon that work. We also had a whole separate other project related to transplant of complex tissues or composite tissues, where you're looking at skin and muscle together and also the systemic circulation. And then we expanded this work using some novel modeling approaches involving hypergraphs in this more recent paper, also in mice. And then we really made the leap to this very important connection to neural mechanisms that regulate inflammation. So for example, this paper in the context of transplantation highlighted that when you do repair the nerve after in this experimental transplant setting, paradoxically, you massively augment the inflammatory response. It's as if the system becomes much more self-aware and the amount of inflammation rises. We also suggested that in the context of the study jointly with Eric Helster's team at Uniform Services University, showing important aspects of the role of the brain in regulating or dysregulating Th17 productions in the context of combat casualty. And as I'll talk about now, our most recent study, also in mice, now talked about perhaps the most crucial inflammation regulatory circuit, which involves the vagus nerve. And again, this is all on, I think, our important development on the way to truly making a realistic and useful critical illness digital twin. But also, I think these kinds of approaches will be useful for any inflammatory or immune disease. So again, for most people here, I probably don't need to explain this, but it's useful to highlight. We're using various tools. Dynamic Bayesian networks allow us to visualize the entire time-dependent data set with highlighting of feedbacks, potential feedbacks in the data. We used a variant of principal component analysis over time intervals called time interval principal component analysis. This other tool, dynamic network analysis, we developed over 10 years ago that allows us, it doesn't really show the feedbacks, but it shows you granular development of networks over time. And then most recently, this dynamic hypergraph tool, which allows us to look at cross-correlated and presumably jointly regulated inflammation simultaneously across tissues, which is essentially a perfect tool set or tool when one wants to think about the potential for neural regulation of the simultaneous explosion of inflammation across tissues. And this study was undertaken with Kevin Tracy, who was the person who put the whole concept of vagal regulation of inflammation on the map, as well as various other crucial aspects of knowledge about the inflammatory response, and Sangeeta Chavan, along with Ashutosh Shah and Ruben Zamora. And so for those that are not familiar with this, the vagus nerve regulates, or is known to regulate, the inflammatory response in a negative way, i.e., so if you block it or if you vagotomize an animal, you get more inflammation. And that's sort of what you can see here. If you were looking at the way that most people have done this, looking at, again, systemic inflammation, and you look at the prototypical inflammatory mediator IL-6, you see that if you compare to a sham surgery, vagotomy generally has higher levels of this mediator in the blood. You also see that in the lung, which again, with some variance, because again, these are animal experiments. But just looking at one other tissue, like say the kidney, already shows you that this is not so straightforward. That yes, there is more, but not so much more as you might imagine. And it's all of it. It seems to be somewhat less than what you're seeing at baseline, suggesting there's some nonlinearity here, something going on that isn't as simple as the vagus just negatively regulates inflammation. And so we thought we could try to learn some things from combining dynamic network analysis and dynamic hypergraphs to look at intra and intertissue interactions, respectively. And that's what we did here. So for reference, inside each one of these boxes, which represent a given tissue like kidney, lung, liver, etc., you can hopefully see interactions that are visualized using our DYNA algorithm. And the way DYNA works is it's looking at cross correlations across time intervals. There are some corrections that happen. I'm happy to address those in the Q&A. But on top of that, we can now use dynamic hypergraphs to look at cross compartmental edges as well. And right now you're seeing everything with black. Our DYNA tool was set up from the beginning to be able to both look at positive correlations, which we visualize with black edges, or negative cross correlations, which we visualize with red edges, which would then correspond to ramping up versus resolution of inflammation, respectively. And as you can see here, this is the sham, this is the vagotomy, not really any major difference, but the picture is and this is the dynamic hypergraphs, but the picture varies tremendously once you do the vagotomy. So if you look at the sham surgery and you look at the negative connections, what you see is a very interesting picture. It's just that there's a very strong in-place network that keeps inflammation in check and under control and not spreading across tissues. As soon as you vagotomize, the majority of those negative connections, negative regulation mechanisms appears. to disappear, meaning that you are you've just uncovered a novel role for the vagus. Not only is it regulating the levels of individual mediators in simple in vitro systems or in systemic circulation, which is what people have looked at previously, but now there's this newly discovered role that says the vagus might be relevant in maintaining inflammation localized to tissues and compartments and stopping it from spreading systemically, which again is the hallmark of major disease. The next thing that we thought about is, okay, if this is going on and it's such a massive effect, is there any kind of a threshold here? Is there anything that we can do with the data to try to discern if we can find kind of where the threshold is and if such a thing exists, and how to look for it? And again, was there a role for the vagus nerve in this process? And the way we did that, and I apologize for the verbosity of the slide, but it's kind of needed to explain this, especially to individuals who don't really have the working daily knowledge of the inflammatory response. But we did, in fact, infer such a threshold. And we did that because multiple tools, multiple analysis approaches suggested that vagotomy differentially affects either the steady state levels or the rate of change of mediators such as interferon gamma, IP10 or CXCL10, MIG or CXCL9, MCP1 or CCL2, and IL-6 across one or more tissues. Importantly, IP10 was a unique edge in the systemic circulation following vagotomy, but not down surgery. And IL-6 was pretty much an output node and not really a significant edge in multiple tissues. But it was elevated, as I just showed a couple of slides back. So ultimately, we came up with this hypothesis that somehow this spatiotemporal arrangement creates a threshold in some fashion. And the way we got to looking at that is we carried out what we call rate of change modeling, just looking at essentially the spontaneous velocity across 2 time points, the rate of change of any given mediator in a given tissue. And we simply plotted that from the least to the greatest. And what you're looking in this graph is essentially a combination of space and time. So each dot represents the velocity, and each word down here represents one tissue or organ, but the order changes depending on the particular mediator we're looking at. And I will guide you to IP10, because what IP10 seems to be doing, right, is it looks like it is having threshold type behavior at baseline, very, very strikingly so, and somewhat less so when you vagotomize, but it's still sort of there with the sigmoidal shape. And really none of the other mediators did that. Moreover, so I mean, that ends up looking threshold-like. But moreover, we can give LPS, and this really highlights how a pro-inflammatory stimulus like that completely disrupts this putative threshold. So this is what we're seeing in the baseline conditions, so in sham versus vagotomy, but with no additional pro-inflammatory stimulus. But if you give bacterial LPS, this whole thing is now linearized. And this suggests that LPS pretty much blows through the role or the negative tonic regulatory effect of the vagus nerve. And again, we had data from our 2018 paper that hinted at this at the time. So this was very striking because it puts out kind of a new concept out there, which is that the body is acting as a kind of inflammation computing mechanism, if you will, and when it's in its intact state, and that pro-inflammatory stimuli, especially potent ones, may exert part of their actions by disrupting some of these thresholds. And the reason we really thought this made sense relative to prior data, so this was all in mice, was that we had also obtained data in trauma patients, segregated, and this was work that Nabil Ajar did, segregated into propensity match cohorts of mild, moderate, or severe injury based on what's called the injury severity score. And then we did dynamic Bayesian network inference, and that kept popping up with this tripartite motif involving MIG, IP10, and MCP1 upstream of IL-6. And as I said, this idea that IL-6 is an output node was seen in the mouse studies. These earlier human studies had already suggested that. And what we did is we said, let's take these data and then the data-driven model and code up a quasi-mechanistic Boolean model that invokes these interactions where there is differential impact on different chemokines depending on the severity of injury, and that each chemokine, previously thought to be essentially redundant, differentially impacts the outcome, especially visualizes IL-6, which is a proxy for severe inflammation, by this mechanism where each chemokine positively regulates itself while negatively cross-regulating the other two. We call this the chemokine switch, in which we were able to, when we encoded this as a quasi-Boolean model, we could reproduce dynamic trajectories, which I'll show in a second. We could predict missing nodes. And importantly, IP10 exhibited a sigmoidal behavior. Now, this was all based on data in the systemic circulation. We did not have access to organ level data in these patients. And this is what I'm showing here. So simulations over here, real patient data over here, and you can see the various mediators. And actually, it all looks kind of interesting, and in fact, somewhat similar between the simulations and the real data, even in subfeatures such as the high variance that you see early on as the system is bouncing around and settling eventually on a trajectory. This is in simulation, and then if you look at the data in the patients, it has much of the same type of look. Now, the one place where we were clearly wrong was when we were simulating IP10. So here's what IP10 actually looked like in our data, which has this sigmoidal shape, but it did not look like that in our simulations. And so that made us think that we were missing something. We called that missing node X. When we included X in the model, you now all of a sudden get that nice sigmoidal shape, as you would expect, because you essentially added a delay. And moreover, we said, okay, could anything in our data be X? And it is, or at least plausibly, which is interferon gamma, which has a similar look to what the node X is predicted to look like. And importantly, it's something we probably should have even known, because IP10 stands for interferon gamma inducible protein of 10 kilodaltons, meaning interferon gamma should be upstream of IP10. But we didn't think of that because normally interferon gamma is considered a mediator that gets produced later. So again, highlighting the power of these kind of tools for discovery as well. Moreover, we had this other paper in 2014 that through a comparison of a cohort of trauma patients without spinal cord injury to another cohort of patients that are trauma patients, but that did have spinal cord injury, it suggested that IP10 is neurally regulated and upstream of IL-10, a sort of prototypical anti-inflammatory mediator, which is essentially the role we inferred for it in the chemokine switch paper as well. So again, all of these are supportive evidence. None of them is the single one definitive proof, but at least it suggests that these lines of evidence are sort of coming together. That while all this was going on, we were also continuing our work on digital twins, and now trying to go after another application, another use, which is how to unify preclinical data and clinical data. And so this paper that I just alluded to from 2015 was based on purely clinical data, but in work with Immunetrics and Jeremy Cannon, who was a trauma surgeon at Penn, we modified that original model from 2015. Simplified it some, but also added the coagulation cascade. So coagulation is intimately tied with inflammation, and coagulopathy is a feature of the response to severe traumatic injury, and also in sepsis. In this study that also involved other collaborators, which we published just recently, showed several outcomes, which I'll talk about in a minute, but also allowed us to leverage a clinical study. This is the prompt study that John Holcomb led originally, and preclinical data from Marty Schreiber's group in swine. And again, this is more of as much as it was a paper about the actual outcomes, it was really more of a proof of concept that we could take, if you will, the best features of a preclinical study, which are, a much larger ability to sample, the ability to perturb, et cetera, but limited in terms of duration typically, because of IRB or IACO considerations. And then the clinical side, which has a different time scale, can't really see a lot of the early events as well. But of course, it's the one you care about because it's clinically relevant. And then combine these two via the data and parameter estimation into a single model that could handle both, and then carry out in silico simulations and studies in both animals and humans. Where, for example, you could take an individual that received a particular resuscitation modality and simulate what would have happened under a different set of circumstances. For reasons of time, I can't really discuss all of it, but I mean, some of the core points are here. Generally, ability to accurately reflect the data, importantly, to predict the outcome and time of death for the human cohort using these models. Some conclusions regarding how certain resuscitation modalities might do in general better than certain others. The idea that earlier resuscitation can reduce both morbidity and mortality. But the important thing also was that the majority of the findings were about how individualized this whole process has to be, which is something that I think the trauma community has been struggling with. And moreover, this was really a proof of concept of something that Gary and I, along with John Bartel, talked about in a paper in 2011, where we talked about the idea of unifying preclinical and clinical data using mechanistic models. And so this idea of this workflow serving as a translational bridge and possibly applicable to other complex disease settings. Now, just to wrap up, ultimately, this is the sort of workflow that we and others have sort of envisioned, where you are, in terms of a rational workflow to address these complex diseases like inflammation, like critical illness, where you're getting high throughput time series data of every level, including cells, mediators, omics, whatever you're measuring. Taking the intermediate step of some sort of data-driven visualization to look at potential feedbacks and interactions, taking those core interactions and coding them up as mechanistic or quasi-mechanistic models, and then mapping those simulations to patient phenotypes. And this is, I think, kind of where digital twins come in. And then now you're making some predictions, or you're having some missing information, which you then need to go forth and repeat the cycle. But also on the other side, we can do rational reprogramming, which of course these kind of mechanistic models lend themselves to, based on the whole field of model-based predictive control, and ideally actuate control. And this is something that we've been talking about and working on, for example, generating a biohybrid device that can help reprogram inflammation. And then most recently in this large-scale grant that's funded by DARPA that involves Gary as well as other people, where we're trying to use this approach and Gary's agent-based models of wounds to reprogram wounds using control policies discovered by deep reinforcement learning, and then bioelectronic devices that act as sort of smart bandages. And so I'll stop there and just acknowledge a very large number of collaborators, and happy to take some questions, hopefully in the remaining time. Thank you.
Thank you for a great talk. I wanted to ask, when we think about collecting patient data to using these digital twin models, and we are trying to predict what happened clinically, are you collecting data that satisfies the start point of your model or the end points, like the clinical outcome, or both? Because the question I feel is always, the data that you're getting, say, from a patient end result, is that a cause or a consequence of the model that you're building? So I wanted to hear your thoughts on that.
Yeah, it's such a very, very astute question. So this is a major vexing concern, because most diseases take years sometimes to evolve. And even sepsis, which is considered very acute, it's very hard to know when the infection truly started. And it's often layered upon something else, like a surgery or some other stay in the hospital that gets you a nosocomial infection that leads to sepsis. This is why we focused on especially severe trauma, because severe trauma, you pretty much know when time zero is. And with the right infrastructure, like we have at Pitt, UPMC, and others as well, you can start sampling very early, which means that you can, in fact, get those early time points. But I agree that for most diseases, that won't be possible, which is why this idea of bridging to preclinical data may be the only kind of way forward, where you need to connect it to the preclinical data, come up with the simulations, have the simulations run out long enough in time to be able to be tested against the clinical data, and at least to some level of validity be considered valid. And then hopefully at that point you can use them for whatever it is you want to use them, whether it's predicting outcomes or designing therapies or what have you. But that's a fantastic question.
Thank you.
So thank you for the talk. It was very interesting, this idea to use AI to figure out some trend in the data and to try to extract prediction. I guess my question is the usual. How do you deal with the fact that you cannot really trace back why the decision has been made? Or if instead are you using some explainable AI technique whereby you can really go back and trace the various steps of the model?
So again, very good question. I didn't directly touch on the exact way we expect to use this type of AI. And frankly, this is why I alluded to the biohybrid device, for example, that we generated. So I think ultimately, that's the way in which something like this could actually be useful, and especially for a rapidly evolving disease process like trauma or sepsis-induced critical illness, which happens, frankly, it probably gets going within seconds to minutes, is quite severe by hours to days. Is the practical challenge even of getting a diagnostic test or a readout fast enough, and even if it was infinitely fast, you might need to wait to hour number 8 or day number 2 to fully lock a trajectory down for an individual, which means no matter what you do, it's Heisenberg uncertainty, to take a licentious liberty with physics. That you simply cannot both observe and manipulate. So that is why we came up with this idea, really pretty much simultaneously with realizing what was inherently challenging about patient-specific simulations. The idea that you would make a device, essentially an auxiliary inflammation control organ, that would be built on, tuned to the simulations, which themselves would be based on the data. But it would begin to immediately reprogram. Pretty much as soon as you could connect it up, it would begin to both sense and actuate to reprogram. And this was funded by the DoD, starting back in around 2008 or so. Because I think this is exactly the point. You need this almost exogenous, near real-time system to be able to reprogram truly acute inflammation. Now, once you talk about autoimmune disease, where you're thinking about a disease that takes years to fully develop, but again, it's a disease that is, most autoimmune diseases actually are characterized by repeated flare-ups. These flare-ups, I think, are so I've been saying that for many, many years. The idea that chronic inflammation is really the chronic or constant restarting of acute inflammation. There's constant positive feedback that kicks in, then it's shut down for a while, and then it comes back up again. The baseline always rises, and so you're left in a, generically speaking, pro-inflammatory state, but the exacerbations are where the real problem is. So you still have that acute component that needs to be addressed, which I think is where the pathology happens, flares. But you at least have a longer time horizon to work in, and that's where the more classical ideas about how to use, kind of reinforcement learning and or model-based control, where you might have either a reservoir of some sort that would deliver, or you would have a warning that gets triggered where a physician would administer something or something like that, right? And of course, cancer is the ultimate frontier in that, and again, also with the longest time horizon. So and with the role of inflammation in cancer, it's an obvious other place to go. But yeah, very, very good question.
Thank you very much. Thanks.
Other questions? I was going to ask, sorry, go ahead.
No, go ahead. Yeah.
I know you have to stop promptly at 4, so I want to make sure we have. I was going to ask you on the experimental side, in terms of monitoring immune response, What are the biggest challenges in terms of sensitivity, multi-factor analysis, and timescale for measurement? If you wanted to have the ideal real-time sensing, say, of all the cytokines, how close are we to that, and what do we need to do? Because whenever I think digital twins, I think actually the sensors are probably the place we are the weakest.
Yeah, so I mean, I see that Gary's quietly there on the call. That's a discussion we have a lot, especially in the context of our DARPA project. I think that is a huge challenge, and especially if you add the dimension that you're going to want to, for many diseases, detect that in distant tissues and organs. That is a fundamental and major challenge. I know of. I mean, there are various indirect ways that have been put out there. There have been interesting measurements in saliva or in exhaled air. I just saw one on measuring cortisol in hair. You could have lots of other ways of potentially getting at some of these. Of course, if you also wanted to add to that cells, and cell populations, not simply inferring them from the cytokine data, that adds yet another element of challenge. I think you've pretty much put the nail on the head that there are that these are major challenges that need to be addressed. Just how to do it. Yep, I agree.
So Gary points out that you can do it in measurements, you can get 12 cytokines in 90 minutes. But for sepsis, you can have an immune state change in 15 minutes.
Yeah, we modified the Luminex platform that we've been very long-term users of to be able to get data in 60 to 90 minutes. But I think that's okay for even for sepsis, but or trauma. It's probably fine. But again, you're looking at getting those data from the systemic circulation. It's not going to tell you really what's going on in the liver or what's going on in the kidney or what's going on in the lung. Yeah.
I mean, I know years ago, Jennifer West was trying to develop these injectable SPR sensors that you could use maybe for transdermal imaging of.
There's nanoparticle type nanoparticle approaches that could glow or show up on some scan if they react with something. I've seen mention of nanoparticles that would also have some sort of ability to do wireless transmission so that you could get. But again, I've not seen any of that reach fruition. Certainly, but so as I said, I think that's a major unmet need. So yes.
Do we have a last question for our speaker? He has to leave promptly at 4 today.
Gary says this is the discussion that he wanted at IMAG, or as I like to call it, every discussion I've ever had with Gary.
Well, Gary, if I'd known you wanted that, I would have been happy to have it with you. I thought it would be dangerous to bring up those topics about feasibility and measurement.
No. I wanted one of the foci of the meeting to be integrating with people who do those types of assay development.
Yes, that's quite a different world of the people who develop those measurement techniques.
Yeah, we need to meet with them.
What we did in the context of that biohybrid device is we, for example, focusing on TNF, and then the idea being that we would, TNF being a core a positive feedback cytokine and can be downregulated by its own soluble TNF receptor. We set up constructs that would be based on multiple NF-kappa B elements, and so then therefore they're sensitive to TNF, and then they would be upstream of soluble TNF receptor. So you can kind of short circuit a loop that happens normally. You could, of course, add to that fluorescence or other types of signaling, or you could get creative and come up with a way to make an electronic signal. There's definitely ways to add to that, but then again, still that's sitting exogenously. Now then miniaturizing that, getting it to go into the body to arrive at places, I mean, that's crossing over right now into science fiction, but I think it could happen, or some version of that is probably how it will happen. So.
Okay. Well, I want to thank you so much. That's a wonderful way to end the conversation with that vision of the future.
Thank
you so much for coming to talk to us. And I'll look forward to seeing people for our next meeting in 2 weeks. And I'll look forward to having you come back maybe and give us maybe more detail on one of these many projects. Thanks so much.
Thank you. Again, thanks to everybody for attending.